Alright, here we are again. Today talking about water, ion, and osmoregulation and excretion. Now, usually when we talk about the excretory system, perhaps our expectation is to only talk about excretion of waste products. However, we're gonna see that the excretory system is so much more complex and has more very important functions for the normal functioning of organisms, of animals. Now, this is again a flipped classroom video, so please do pay attention and take notes and come prepared to take a quiz during our next lecture. Well, as before, I divided this lecture up into different sections, and as before, I'm going to start with evolution as the number one topic. Then we're going to move on to the different functions of excretory system. We're going to talk about the kinds of challenges that terrestrial animals are facing, and then we're going to talk about the different challenges that aquatic animals are facing with regards to their excretory system. Then, the fifth point, we're going to talk about different types of excretory organs that we find in different animal phyla. Then, number 6, we are going to talk about kidney function. Now, every time we go through a lecture, there's always going to be this one section that's going to be longer, that's going to be a little bit more difficult, that's going to be a little bit more conceptual, and that is section 6 for the lecture on excretion. And then finally, we close this lecture by talking about different types of nitrogenous waste. Now let me start with evolution, and I really do not want to talk too much about this point, even though it is insanely interesting. But I want to read a quote to you from a publication that I have used before in this class. Remember when we talked about the origin of the sensory system, we learned that sponges have certain patterns of gene expression that we also find in sensory systems, and that some of these patterns of expression are particularly related to choanocytes. Alright, let me read this. A still less expected set of commonalities is evident between vertebrate sense organs, such as the ear, and kidneys. Both sense organs and kidneys express the same suite of regulators in development, and there are a number of diseases that affect the ear and kidney, in particular leading to the biomedical term otic renal complex. Common attributes of distinctly different organs are often dismissed as co-optation, but this is too easy. How such co-optation occurs is critical to understanding evolution. We argue that, whether considered co-optive or not, they likely reflect some aspects of shared ancestry, and that such common origin may be supported by examination of the basal lineages. In this unexpected case, the commonality of kidneys and sense organs, we argue below that this could reflect cellular organization in sponges, in which groupings of choanocytes may serve multiple functions and subsequently evolved into the sense organs and kidneys in bilaterians. Whoa! I know that at least one of you has lots of questions at this point, and to be honest, I have a lot of questions too. So we're not going to really try to answer them at this point, This could be a term project for comparative animal physiology. But let's just take this, to ponder a little bit how evolution works. Going from a sponge that has no true differentiation into tissues, providing the raw material to produce two completely functionally different systems, the sensory system and the kidneys. And we're going to see in at least one of the excretory systems in one phylum that there are cells that again remind you of choanocytes, that even have flagella. Now we're going to come back to that later. Pretty cool stuff though, isn't it? So let's turn our attention to function, since there is so much we have to cover in this one lecture. Now, as the title of this lecture suggests, there are 4 different functions of the excretory system: the regulation of water volume, the regulation of ion concentration, and the regulation of osmolarity, in addition to the removal of wastes, which is what we usually think of when we think about the excretory system. Now we're going to see that all of these are interrelated. So water volume does depend on ion concentration. Osmolarity depends on ion concentration and the concentration of certain waste products. So all of these in many ways are related. In terms of regulation of water volume, now this is something that we usually do not think of when we think about the excretory system. And we're going to see a little later that for aquatic organisms this is really a critical function. Now we have to remember that water is a major portion of an animal's body mass. So most of our bodies actually are composed of water, which is the stuff that dissolves a series of salts, proteins, other solutes that are important for all sorts of chemical reactions in the body. Water is also a major transport vehicle. We just finished talking about the circulatory system. Now, obviously, blood is, to a large degree, water. Now, when an animal becomes dehydrated, therefore, we are going to compromise the circulatory system. We are going to compromise the concentration gradients in different ions that are important for maintaining life processes. And we're also going to compromise the regulation of body temperature. So water volume is very important. Now, in terms of regulation of ion concentration, we have to remember that many of the solutes in our body, such as sodium and chloride, calcium, are crucial for so many functions. Just think about sodium and potassium during the transmission of an action potential in nerves, or the propagation of a depolarization front in the heart that lead to the contraction of the heart to pump the blood through our blood vessels. These are life-sustaining processes that depend on the correct concentrations of different solutes. So an imbalance in electrolytes, an imbalance in these solutes can impair all those functions that depend on specific changes of the membrane potential. It can disrupt intracellular processes. Just think about calcium, for example, which is an important intracellular messenger. Well, without the calcium, signal transduction pathways, or many signal transduction pathways, would not work properly. And finally, and this is obviously also related, the osmolarity. Water moves down concentration gradients. It moves from low concentration of solutes, which turns out to be a high concentration of water, to compartments with a high concentration of solutes, which inversely means a low concentration or lower concentration of water. So osmotic pressure is going to be very, very important. And of course it depends on many things. Many of the solutes we already mentioned, but also proteins in the blood, for example. Now, changes in salt concentration in one compartment, be it the intracellular environment, the cytosol, be it the interstitial fluids, will lead to movements of water between compartments. Now, in extreme cases, when too much water moves in or out of cells, cell death may occur because of a rupture of the plasma membrane. Now, in terms of osmolarity, now maybe just a little reminder, you probably have seen this in chemistry and remember it way better than I do, but we just have to remember what osmolarity is referring to. It is referring to the number of dissolved solute molecules per liter. So let us consider as an example a solution of sodium chloride that is 150 millimolar. When we dissolve this solution, the sodium and chloride will dissociate and create two dissolved particles for each NaCl. That is 150 millimolar sodium chloride solution is equivalent to a 300 milliosmolar per liter solution. It's equivalent to a 300 milliosmolar solution. Okay. All right. Now, here are some numbers that we've already tossed around during our dissection of the fish, where we already talked a little bit about the excretory system. So when we think vertebrates, most fish and other vertebrates are around 225 to 400 milliosmolar per liter. Now let's compare that to fresh water, which is usually less than 25 milliosmolar per liter, and seawater, marine water, that is usually around 1,000 milliosmolar per liter. As you can see, there are going to be strong osmotic pressures acting on especially aquatic organisms, as we're going to see shortly. Alright, and finally the removal of wastes, which is what we usually think of. When we think about the excretory system, we have nitrogenous wastes. We deal with those in the last section of this lecture. We have different types of solutes that may be present in excess and our body has to get rid of them. Chloride, sodium, potassium, for example. We have creatinine, which is derived from creatinine phosphate, an important molecule in the cells that provides for rapid substrate level phosphorylation when ATP is needed in the cells. There are other dissolved ions, inorganic and organic compounds, small proteins, hormones, and all sorts of different metabolites that we also get rid of through the urine. And as I mentioned before, the excretory system is also important in regulating acid-base relations inside of the blood by removing an excess of hydrogen ions or an excess of bicarbonate ions or by perhaps reabsorbing these ions when needed. Alright, let's move on then to the third section. We'll be talking about the kind of challenges that the excretory system is facing in terrestrial animals first, and then we talk about aquatic animals. Alright, as was the case with the respiratory membrane, desiccation is the biggest problem for terrestrial animals. Now, there may be all sorts of different solutions to prevent desiccation. We're going to mention a few. For example, marine birds and reptiles have salt glands to get rid of the excess of sodium and chloride that they ingest with seawater as they are feeding. There are different ways also to reduce obligatory water loss. I'll explain that term in just a minute. And there are also ways for reducing evaporative water losses. Alright, so let's cover these one by one. First, salt glands. Salt glands are able to concentrate sodium and chloride so that we can get rid of these solutes without getting rid of a lot of water. And that is important. You may have heard many times before that, if you suffer a shipwreck and you're out floating somewhere in the ocean and you feel really, really, really thirsty, never, ever drink seawater. Because of its high osmolarity, it actually will require more water to get rid of the excess of salt than you actually gained from drinking, which means you're excreting more than you are ingesting, and that of course means you're gonna dehydrate. Now, these salt glands use a countercurrent mechanism to continuously move sodium and chloride into the lumen of the salt glands with very little water to get rid of this excess of salt. Now, the salt is usually actively transported from the interstitial fluid to the lumen of the salt gland, but we always will have a positive gradient that is a higher concentration at any point during the salt gland in the blood vessels than in the lumen of the salt gland, which means there's going to be a continued movement of sodium and chloride into the lumen. There's not going to be any back movement due to an upset concentration gradient. Now, as you can see here, and I'm going to just need a little fake laser pointer. Here we are. Here we have a turtle, a marine animal, a marine reptile that seems like it is shedding a lot of tears, like it is really, really sad. But it's not. It's actually shedding the excess of salt that it has ingested with very, very little water. And the same is true for this penguin that has these salt glands right above the ears, right above the eyes, okay, that drain this highly concentrated solution of sodium and chloride through the nostrils, and it's lost as little droplets. Now, one of my favorite organisms to use salt glands to get rid of the excess of sodium and chloride is the marine iguana from the Galapagos Islands. Now, this reptile, it's a weird reptile for many reasons. It's fascinatingly weird. Actually feeds on marine algae in cold water. Now, as it is feeding, it will ingest a lot of seawater. It will start to get cold. It has to come out of the water to warm up on land again. and at the same time it will use its salt glands to concentrate the sodium chloride. And then when you watch them for a little bit as they're warming up in the sunlight, you will make them a very jerky movement with their head, a little snot, and out come salt crystals. I mean, this is almost crystallized salt that comes out of their nostrils. I mean, really, really cool animals. All right, so let's talk about obligatory water loss. Obligatory water loss is loss of water from the body that is required for the body to perform normal functions, to have a normal metabolism. And there are three sources of obligatory water loss. The first one is through breathing. Okay. Now, why do we lose water when we breathe? Well, you can certainly make sure that you're losing water as you breathe if you're standing in front of a window and you just blow a little air on the window and you see condensation forming. That condensation is water that came out of your lungs. Your lungs are, remember, 100% saturated with water vapor, and that was necessary to maintain the respiratory surfaces moist, and that was necessary for the proper diffusion of oxygen and carbon dioxide as they move across the respiratory membrane. So every time we breathe out, we're losing water. But it's necessary. I mean, we need to breathe out water because we need to have a 100% saturation of the air in our lungs. Now, different animals have different mechanisms to go about it. Many in their noses, very specialized noses, have mechanisms to cool the air rapidly as it moves through the nose. And guess what happens when the air cools? The water will condense, and it condenses on the inside surface of the nose, where it can be taken up again into the body. The saiga antelope, for example. Yeah, Google that. Saiga antelope. There was just recently a little photo report on the BBC.com about it. Has these weird nostrils that do exactly that. They cool down the air in the summer so that they can retain water, and those nostrils will actually warm up the air during inhalation in the winter to make sure that the cool air doesn't get into the lungs and then could potentially lower core body temperature. So there are a variety of mechanisms to cool the water. We have obligatory water loss in urine. That is, we cannot get rid of salts and of urea and all these other goodies in the urine if we don't use water. Now, as I say this is not entirely true. There are some animals, desert animals for example, that are able to concentrate their urine so much that they hardly lose any water with it. And finally, there's an obligatory loss of water in feces. Now, feces to move through our intestines need to have a certain content of water. You may remember, and we're going to talk about this a little bit more in the next lecture and during digestion, the large intestine is very important in regulating water as well, to make sure that we're not losing an excess of water in our feces. But we do need water to move the feces, to get the feces out of our bodies. Now, some animals have achieved a very dry feces. They can get rid of it by almost recuperating all of the water in them. Okay, now one additional note about obligatory urinary loss. It's very interesting to see how diet can influence kidney function. For example, well, think you eat a big steak or sushi or fish or any high protein content meal. Now that high protein content meal is going to generate a lot of nitrogenous waste because we cannot use or store all of the proteins that we are ingesting. So we deaminate the amino acids, and that generates nitrogenous waste, and we use the rest of the amino acid to convert into either carbohydrates or lipids. So a high protein meal will lead to high nitrogenous waste production, which will require a high rate of blood filtration in the kidney. So your body will want to get rid of all this nitrogenous waste, and you actually do oftentimes feel very thirsty after a high protein meal. Now, one exception to the inability to store amino acids are polar bears. Now, they don't eat for months, but they continue to make nitrogenous wastes. But then, instead of getting rid of these wastes, they have a very handy ability to recycle the nitrogenous wastes back into amino acids and protein. So they're recycling it. Now there are a variety of bacteria and microorganisms that will do the same thing. And finally, let's think about the vampire bat that we talked about so much during our last field trip. It's a very, very cool and interesting bat. It consumes large volumes of water with blood. Water is heavy, and for an animal that depends on flight, it's not convenient to be heavy. So they have evolved mechanisms to very quickly excrete a great amount of water while retaining most of the nutrients. Yeah, just cool animals all around, those vampire bats. Alright, and then finally we also have the problem with evaporative water losses. Humans sweat as we feel hot. It's a mechanism to reduce body temperature. And of course, the hotter it gets, the more we will sweat, the more water we will lose. The water in sweat is hypoosmotic to the blood. Hypo meaning it has an osmolarity lower than the blood. And that means that the water that's left behind, or the fluids that are left behind in the body, now have a lower volume, but a slightly higher concentration of solutes as well. So we need to, in many ways, find mechanisms to reduce this water loss. Now, don't forget that sweating is a life-saving adaptation. Maybe the best way is just to drink your water and stay hydrated all day long. Okay. Finally, in this section, an additional note about metabolism. Now, metabolism is the sum total of all the chemical reactions in the body, and of course some of those reactions we've already learned about during Biology 1, and I'm talking about cellular respiration. Cellular respiration, beginning with glycolysis, breaks down the carbohydrate to extract ATP. But you may also remember that in these reactions we formed water and carbon dioxide as side products. Now this water actually comes in handy for different animals, really for all animals. It's a way of, I don't know, you could say of internal drinking. You're generating the water yourself. Now there are some animals that almost completely rely on this metabolic water for their survival, like some of the desert mice that I've already mentioned. And other animals that are dealing with issues of accumulating too much volume of water, well, they need to find ways of getting rid of this excess metabolic water. Okay, now we move on to the next section, aquatic animals. Now we talked about many of the issues concerning the excretory system in lab, but it's worthwhile to go through them again here in class. Now, as mentioned previously, fish by and large have impermeable skin to the different salts, water, etc. But of course their thinnest membrane that is important for gas exchange, the epithelial tissue of the gills, is very vulnerable to the uptake or loss of water as well as salts. Now it turns out that through gills, mostly gills, marine animals tend to gain salt and lose a lot of water, and freshwater animals lose salt and gain water. Okay, why is that the case? Now I should say I may ask you that question again sometime soon, so think about it. Alright, freshwater fish gain water and lose salt when ventilating gills. Now that means that the fish need to get rid of an excess amount of water, or else they just inflate like a balloon and, I don't know, maybe burst at some point in time. So the freshwater fish need to produce large amounts of very dilute urine. Now it turns out that, of course, the concentration of sodium and chloride in the fish is higher than in the freshwater, so they tend to lose sodium and chloride to their environment. Now it turns out that for that the fish also have a solution in the form of so-called chloride cells in the gill epithelium that will transport sodium and chloride from the water continuously into the fish's capillaries. So through the gills, through the fine epithelium, the fish loses the salts, but it also takes up salts continuously, actively expending ATP through the gills continuously. So here we can see on this figure, and I need my pointer again. Where did it go? Yeah, there it is. Alright. Can see that the fish is going to have the problem of an excess of water coming in through the gills by osmosis. There is some salt and water that comes in through the food, through the mouth, but freshwater fish do not drink. There is the active uptake of sodium and chloride across the gill epithelia into capillaries via the chloride cells. And obviously the production of very dilute, copious, that means like just very large quantities of urine containing very little salts. Now let's briefly contrast that with saltwater fish that gain salts and lose water across the gills. Well, they need to produce very little urine and highly concentrated. They do drink seawater to replace water lost, and they expend energy to transport excess salt out of the body through the gill epithelial cells. So whereas the freshwater fish expended energy to pump sodium and chloride in, these fish are pumping them out. And now, if you think about it, it makes perfect sense why a freshwater fish would do awfully in marine water, and why a marine fish would do awfully in freshwater, unless you are adapted to swim in both. Alright, so here on the graph, on this figure, we can see the fish drinks water, obtains salts and water from food. Water leaves the gills, gill capillaries, enters the seawater. There is a heavy active excretion of sodium and chloride across the gill epithelia, which maintains also the osmolarity at around 300 milliosmolar per liter, and very small amounts of concentrated urine containing large amounts of salt. Okay, so very different environments require different approaches to deal with these problems, and the excretory system plays a central role, but so do the gills. Not just the kidneys, so do the gills. Now there is another group of fish for which I do not have a slide here, and that is sharks. Now sharks are interesting because their body osmolarity is about 1,000 milliosmolar per liter, which, if you remember, is the same as seawater. So they are isoosmotic with the seawater, which means that they do not lose any water to through their gills into the ocean. Now, if we look at the composition of solutes between seawater and the sharks, they are very different. It turns out that sharks increase their osmolarity by increasing the concentrations of urea in their blood. Wow, so they actually maintain or keep a lot of urea around in the blood so that they are isoosmotic. But then you might think, well, geez, urea in the blood, well, that can't be all that healthy either, right? Because after all, urea may help denature proteins. It may cause a lot of problems as well in high concentrations. It is, after all, it is toxic. Now, in order to deal with the excess urea, the sharks produce a compound called trimethyl oxide N-amine, TMOA, trimethyl oxide N, letter N, amine. And this compound does what? Well, it neutralizes the urea, the negative effects of the urea. Pretty cool, huh? Alright, now up to this point we've talked only about vertebrates, and this is true for terrestrial as well as aquatic animals. However, in the animal world, the vertebrates would fall into a group that we call the regulators. That is, they regulate their internal environment continuously, keeping salts out, water in, or vice versa. They're constantly keeping those numbers in check. But some other animals actually we could consider conformers, in the sense that they allow their internal environment to change along with the external environment. Alright, now what is the advantage of being a conformer? Well, regulation requires a lot of energy. If you are a conformer, you have to deal with exceedingly high concentrations of some solutes, but at least you don't expend much energy to maintain your internal conditions. So just remember, there are regulators and there are conformers. Regulators have a heavy cost associated with regulating their internal conditions. Osmoconformers then match osmolarity of blood and other fluids to seawater at 1000 milliosmolar. And here we have a lot of marine invertebrates and a few marine vertebrates. So because they change along with the conditions of the water, they tend to lose or gain much less water than regulators. As mentioned, they expend less energy. Now, they may still have ion concentrations that are similar to those of osmoregulators. Now, what these fish are doing is they're allowing their osmotic pressure to change, but they do so without changing the concentrations of specific solutes such as sodium chloride and potassium, which are critical for the functioning of so many processes, as mentioned earlier. Now, the body fluids tend to be less salty than seawater, so they tend to gain salt. Well, they still have to regulate the salt, and they do so usually by using a rectal gland. Now, sharks do that, by the way, as well. Alright, so on to excretory organs. Now, all excretory organs, or most organs, have the following in common. They filter the blood. After filtration, there may be some reabsorption of all sorts of goodies, and the process ends with secretion, that is, we're getting rid of solutes and nitrogenous wastes and whatnot. So that is something that we're going to see in all the different excretory organs. And that is exemplified here by this figure where we have blood flow. An artery, an arteriole comes in. It is a fenestrated capillary here. Through these fenestrations, water is allowed to exit along with small solutes, but not proteins. Not proteins, nor cells. If cells or proteins make it in here, it's because there is something wrong with your kidney. Then this original filtrate then is subject to reabsorption of certain components of the urine that the water wants to retain, and there may still be some secretion into the lumen of the excretory organ. So this is our generalized kidney. Now, how does it play out in different animal phyla? Well, first we're going to start with the simplest form of kidney, the protonephridia. And that is something that we find, for example, in flatworms. But even here we do have filtration into a set of branching tubules that then will drain and be secreted. Now, the filtration requires, in these flatworms, ciliated cells. I'll show you the picture in just a moment. Any solutes that need to be reabsorbed will then be reabsorbed, and then eventually everything will be dumped into the environment through openings in the body wall called the nephridiopores. In terms of nitrogenous wastes, well, those diffuse right out of the body. Now here's that picture. Now remember the note on evolution and the fact that kidneys may be derived from choanocytes. Well, here we have our specialized cell with these cilia. The beating of the cilia is important to generate a current that will suck in fluids through these slit-like openings. Not everything can move through these openings, so the contents are selectively filtered in this structure. Now the ciliated cell is referred to as the flame cell. Alright, and this whole thing here is the protonephridium down here. Then all of the fluids that are to be secreted will end up here in this tubule that goes out to the nephridiopore. Alright, all elements are there. Let's move on to the metanephridial system, which is a system that we find, for example, in earthworms and other annelids. Now, each body segment has a pair of metanephridia. We're going to see that these consist of a tubular network that begin with a funnel-like structure called the nephrostome. From there, coelomic fluid is collected, and the coelomic fluid contains nitrogenous waste, among others. They are moved through the tubular network until they are finally excreted, the contents, through nephridiopores in the body wall. Now, throughout the tubular network, sodium and chloride may still be reabsorbed. So something that we've seen before and in our generalized schematic of an excretory system. So here we are. We can also see there is an extensive bed of capillaries around the tubular network. We're going to come back to these blood vessels in the human kidney or the mammalian kidney shortly. So somewhere in here we have our little funnel. It's not quite visible. Probably here. Yeah, this is it, the nephrostome that collects the coelomic fluids, moves it through the tubular network to reabsorb any goodies that we need to hold on to, and then finally the contents of the stuff that they don't want to hold on to move out the nephridiopore. Alright, now we're moving on to a different type of excretory structure that deviates somewhat from the generalized one that we've seen apply before. So now we're talking about Malpighian tubules. Those are tubules that go directly into the hind gut, so, or we could say they emanate from the hind gut. Now what's different about Malpighian tubules is that they do not use filtration. There's no filtration. All of the solutes that need to be removed are actually actively transported and secreted. So solutes, uric acid, all of these waste products are transported from the hemolymph into the lumen of the Malpighian tubule. Now that creates an osmotic gradient. The inside of the tubule will have a higher osmotic pressure, and that means that water will follow. So water follows passively. Solutes actively, water follows passively. Now from the tubules, the contents will move into the hindgut, where we may still see some reabsorption of water and several solutes, but nitrogenous waste and any other solutes that are no longer required are excreted together with feces through the anus. So here we have a picture that shows what this looks like. Where is my pointer? Here it is. Here is the intestine, the hindgut. Here are the Malpighian tubules that do not filter the hemolymph but actively transport solutes into the lumen. This increases the osmolarity inside of the lumen. Water follows down its concentration gradient passively. From here, all the contents are discharged into the intestine, where salt and water may be reabsorbed, but nitrogenous waste and feces are expelled together through the anus. Alright, so finally let's talk about the kidney as we find it in vertebrates. We're going to see that this network of tubules is highly specialized and complex, and that epithelial cells actively transport sodium and other ions for salt and water homeostasis and nitrogenous waste elimination. There's still a lot of movement across the epithelial cells inside of the kidney. So what does the kidney look like? Now here we have a schematic of the excretory system. We have our kidneys, the blood supply coming in from the artery. We have veins exiting the kidneys. Then we have a ureter that will take the urine down to the urinary bladder, where finally through the urethra it will exit our body. Let's look at a close-up of the kidney. We can actually see that there's some structure to it. There's an outer renal cortex that does look very different in cross-section. And then we have a medulla just beneath it. Now from the medulla we're gonna see these tubes, these ducts entering what we call the renal pelvis, where the urine is then drained through the ureter down into the bladder. So we have a renal cortex, a renal medulla, and a renal pelvis. And that brings us to kidney functioning. Yay! Alright, here we are. The fundamental structure, basic structure of the kidneys is the nephron. It's the functional unit. Now, there may be several million of those in each kidney. Now, each nephron consists of a renal corpuscle where the blood is filtered, and then a network of tubules where secretion and reabsorption is occurring. Finally, there's going to be a collecting duct where, in the end, still many changes may happen. So here we have a picture of a nephron. Here is the renal corpuscle, which consists of Bowman's capsule, which is this structure, part of the nephron, and it also consists of the glomerulus, which is a small network of capillaries from whence the blood is going to be filtered. From here, the filtrate will go through a proximal convoluted tubule. From the proximal convoluted tubule, it will move down into this long structure here, which is called the loop of Henle. The loop of Henle has a descending limb, which is this side, where the filtrate is moving down, and it has an ascending limb on this side, where the filtrate is moving back up. We have in the loop of Henle thin sections and thick sections. And all of these ducts here of the loop of Henle are accompanied by a network of capillaries called the vasa recta. From the ascending limb of the loop of Henle, the filtrate will move through the distal convoluted tubule, from whence it will move into the collecting duct, from where it moves down towards the renal pelvis. Yes, you do need to know the structure of the nephron, so do remember and be able to label a figure of the nephron. Alright, so as an overview, in the renal corpuscle, filtration is going to take place. In the proximal convoluted tubule, we have selective reabsorption. Things such as glucose that we don't want to lose through the urine is going to be reabsorbed. The same holds true for many amino acids and salts. The loop of Henle is important to establish a gradient in the kidney, as I'm going to explain shortly. The distal convoluted tubules are the site of additional regulation of several solutes. And then finally the collecting duct, where still reabsorption of water and solutes may occur. I'm going to talk about each one of these components of the nephron and what actually happens there. Alright, the renal corpuscle. This is where filtration is going to take place through fenestrated capillaries and through specialized cells of Bowman's capsule, specialized cells that are referred to as podocytes. Now the blood comes into these capillaries by an afferent arteriole. Now that's a term that we have already seen twice now. Afferent meaning going towards or coming towards, in this case, the glomerulus, and then it is drained by the efferent or outgoing arteriole. So here we have a figure with a structure. Bowman's capsule consists of a epithelial cell layer. The cells that surround the capillaries in here are the podocytes that have slit-like openings. And here we have our afferent and efferent arterioles. Now let's have a look closer at these blood vessels and podocytes. You can see that the capillaries are fenestrated, so they have little holes, and that the podocytes have slits that allow for the movement of solvents, that is water, and small molecules. Now from Bowman's capsule, the filtrate is going to enter a tubular portion of the nephron, in particular the proximal convoluted tubule, from where we move through the loop of Henle, the distal convoluted tubule, and then into the collecting duct. Okay. Now, in terms of filtration, as blood flows through the glomerulus, about 20% of the plasma leaves the capillaries and filters into Bowman's space. 20% of the blood in the capillaries actually exits the blood vessels. Now, small molecules, organic molecules, and solutes, well, those will filter right through, but large proteins and cells stay in the blood vessels. Now we call this the glomerular filtrate. This is the stuff that filters directly into the glomerulus. Now when we want to measure the rate of filtrate production, we are talking about the glomerular filtration rate, or how much of the blood is going to be filtered per unit time in the glomerulus. In the proximal tubule, we see reabsorption of many molecules. Now first, the filtrate is around 300 milliosmolar, which is almost isoosmotic with the blood. Now at this point, however, we're going to reabsorb solutes. Many of those, such as sodium, are reabsorbed actively, and water follows passively. At this point, we're also reabsorbing sodium potassium chloride bicarbonate for acid regulation of the blood. Organic molecules such as glucose or amino acids, all those are reabsorbed. Alright, now solutes and water are reabsorbed in proportion to each other. What I want to say here is that as the solutes move out and the water moves or follows passively, the osmolarity of the filtrate in the proximal convoluted tubule remains at a 300 milliosmolar per liter concentration.